The metabolic phenotype varies widely due to external factors such as diet and gut microbiome composition. The hypothesis brought forward today is despite these temporal fluctuations, urine metabolite profiling studies have suggested that there are highly individual phenotypes that persist over extended periods of time. This was then tested by the analysis of human breath within a group of subjects during 9 days by mass spectrometry. Mass spectrometry is the science of displaying the spectra of the masses of the molecules containing a sample of material. For this analysis, the 11 subjects, consisting of 6 males and 5 females, were asked to breathe through a heated Teflon tube connected to the curtain gas port of a quadruple time-of-flight mass spectrometer. The sampling tube was surrounded with a heating tape mechanism, tempered at 90 degrees Celsius to prevent water condensation and to minimize the losses onto the walls of exhaled compounds. This process was performed in triplicate, which is a set of 3. The exhaled breath encountered an electrospray plume, where some compounds get ionized and subsequently mass analyzed. In this process, only the last few seconds, which was normally around 6 seconds, of each exhalation was considered, excluding from the analysis the first part of the exhalation, which reflects mostly dead volume in the upper respiratory tract. Figure 1 represents the real-time analysis for the rapid breath printing of subjects. During the 1.5-hour experiment, the 9 subjects breathed into the mass spectrometer. The subject label codes are indicated at the tip of each of the 3 replicate measurements in the top trace. The 3 traces correspond to the ion intensity as a function of time for MZ 59, 151, and 207. Figure 2 represents the 3 representative ions from Figure 1. The box plots provide an overview of the intra and inter-subject variability during the whole period. The central mark of each box corresponds to the median. The edges of the boxes are the 25th and 75th percentiles. The whiskers extend to the most extreme data points not considered outliers, and outliers are plotted individually. Figure 3 represents the projection of the 193 breath mass spectra onto the first 3 dimensions obtained. The inset displays the blind classification results upon a cross-validation. The overall breath mass spectrum to breath donor recognition can be seen at 76%. Based on the real-time mass spectrometric analysis of exhaled breath of a group of subjects during 9 days, it is concluded that the intra- and inter-subject variability for some compounds detected in breath can vary across the board. It was also established that the breath analysis may contribute, together with other techniques, towards a future individualized oriented healthcare.